Okay, so here we have our bacterial cell that has the plasmid in it that we've inserted our gene of interest. And here we said, for example, we were saying that the gene of interest was the gene that produces insulin. So here they've called this a recombinant bacterium. Another way to say that is transgenic. That's the word that we've been using. Okay, so transgenic organism, or in this instance specifically transgenic bacteria. So this bacterial cell is now going to be grown in culture. So that just means that it's grown in a little dish with some liquid, and in that liquid it has all the food and nutrients and everything the bacterial cell needs to grow, and the bacterial cell is going to divide. Every time the bacterial cell divides, that plasmid divides along with it, and because we have that insulin gene in there, that's going to divide also. Okay, and so then what will happen is that gene will be expressed. So the cell will go through transcription and translation with that DNA, and that DNA sequence will be read just as if it's bacterial DNA, and so that bacterial cell can produce human insulin. So that was the example that we were using, is that this gene here is the gene for human insulin. So now we have a bacterial cell producing human insulin, and then we can harvest that human insulin and we can use it. And so that's actually how insulin is made, that you would buy today or use today if you were a diabetic. It used to be that insulin was taken from cattle. And so you would be using cattle insulin if you were diabetic. And so I'm sure you can imagine that might cause issues, that you're not using actually human insulin. So now we can make bacteria that can produce human insulin, and so that can be used. And so obviously you're using human insulin and you're not going to have any problems. Okay, so we can also use that protein. So here we're going to harvest the protein. So if it wasn't insulin, some other examples, we could do some basic research on the protein. Another protein that is produced in this exact same way, like the insulin, is human growth hormone. Or we can put a gene in that makes a special protein that dissolves blood clots in the heart. Or if we don't want the protein, we can then use the copies of the gene that are made to do something else. So remember, every time the bacterial cell reproduces, it's copying that gene of interest. So this is a good way to just make copies of a gene if you want to do something with the gene. So we could do basic research on the gene, or some other examples here include the gene being used to alter bacteria for cleaning up toxic waste, and that is done. Or the gene for pest resistance is inserted into plants, and that's also being done. Actually, we're going to look at that next.